questions related to my research path, my other professional experiences, as well as transitioning into and out of academia for anyone who is interested in pursuing alternative career paths. Ultimately, I hope the attendees today leave the talk with insights into navigating a path or career in academia, and a career in academic research focusing on psychoactive substances. I would love to stimulate some discussion, of course, around the use of and acceptance of psychoactive substances in society and in medicine. And ultimately, I'm happy to connect with you, with interested individuals looking for opportunities for cooperation, collaboration, and growth. So finally, if at all, if there's any point in time where I'm speaking too quickly, or if you'd like for me to reiterate something, please, to the organizers, feel free to interrupt. Additionally, if anyone has any questions, you can, of course, submit them in the Q&A at the end, I believe. Also through, I think, the Q&A feature in Zoom. I am also available to answer questions through email. I have my email linked on the final slide, and I'd be happy to set up some time to chat or communicate through email. So I'm going to start with the abridged version of my origin story. My early fascination with neuroscience and anything brain related started in my undergraduate training, as most do. And I knew at the time when I first discovered my passion for the brain and brain science that I would need experience working in a laboratory to ultimately pursue graduate level research and then a career in academia. And, although this is not a stated requirement for many programs, it certainly helps to have the applied experience. And so if I can encourage anything today with the trainees on the call, one of the big takeaways I think I'd like to express is that find opportunities to really dive into something, acquire that valuable experience, but most importantly, really test your passion and test your resolve for your passion pursuit. I think you can best know if you like or not like something by, of course, involving yourself directly in it. So I volunteered in the lab at Western University, as depicted here in the photo on the slide, with my supervisor, Dr. Suzanne Schmid, for 4 months. And then I went on to continue a 4th year honors thesis project for another 8 months in that lab. So a total of 1 year in that lab. To this day, I communicate regularly with Dr. Schmid. And importantly, another takeaway for the trainees is really consider building a strong professional network. You will be supported by them. They will be your champions as you develop professionally, and you will be stronger for having a large support group or support network behind you. So ultimately, the research that I performed as part of my undergraduate degree was published in the journal Genes, Brain, and Behavior in 2013. You can access the paper by searching the PubMed identification number at the bottom of the slide. I'm not going to go into great detail about this research, but I would like to say that this training opportunity afforded me experience in running behavioral tests, in using animals for research, and in preparing drug injections and acquiring wet lab experience. So it really laid the foundation for ultimately the rest of my training and my experience to date and where I'm at currently. So after completing my undergraduate degree, I had a greater resolve as I tested my passion for academia and research, and I decided to expand upon the skill set that I had developed through that experience in Dr. Schmid's lab by pursuing a Master's of Science degree. And so I was accepted into the program at the University of Edinburgh in Scotland. I have a couple of photos here depicting my fun times with my cohorts and spending some time at a wonderful neuroscience conference in Italy. Big takeaway here again for any trainees on the call. Seek opportunities to expand your skill set and build upon what skills you no doubt already have. So for example, I had acquired skills with behavioral tasks and working in a lab as part of a team in a lab. And with this opportunity at the University of Edinburgh, I really wanted to complement my skills in behavioral tests with tests in neurophysiology and running in vivo electrophysiological recordings. So I joined the Memory in Space Laboratory, which is supervised by Dr. Emma Wood and Dr. Paul Dudchenko. And I went on to complete a project that examined place cell function in relation to spatial learning. So I'm not going to, again, dive into greater detail on this research output either, only because I could likely spend the entire talk today on this paper, but I would encourage anyone interested in understanding more and finding out what we did to search that PubMed identification number at the bottom of the screen. The work was published in 2016 in the journal Hippocampus. So following my master's degree training, I had a number of opportunities in front of me to continue pursuing an academic path, an academic career. But I wanted, excuse me, I wanted to challenge myself. I wanted to challenge my resolve outside of an academic pursuit. I was familiar with what was in front of me for academia, but ultimately I wasn't aware of what opportunities existed outside. And so I returned to Canada and spent nearly 5 years working for a number of different nonprofit organizations that included science communication, science education, as well as different research funding agencies. And so I really diversified this skill set that I had been expanding towards a more holistic perspective of what it meant to be a neuroscientist and a neuroscience professional. At this time, I also co-founded the Toronto, Canada chapter of NeurotechX, which is, as mentioned before, the largest community for neurotechnology enthusiasts, and I currently work there as the Connectome Manager, overseeing the Global Chapters Initiative, which is now up to 30 chapters strong. And it's actually through this work that I was invited, of course, today for this opportunity. So a huge thank you again to the organizers, to all of you, the attendees, as well as anyone involved with NeurotechX locally there. Takeaway that I'd like to have for most of the trainees or anyone really on the call with this slide is that if you are considering a change, maybe not necessarily leaving academia, but a change within your comfort zone or what you're used to traditionally, I find it personally quite valuable to take a step outside. You'll grow. Again, you'll acquire this kind of holistic perspective of neuroscience. For example, I learned many project management strategies that I see as currently uncommon in academia through these opportunities, and it'll kind of make you a stronger candidate for the next thing that you embark upon, like my PhD, which brings me to where I am at now. So recently I returned to academia as a PhD candidate and to basically incubate the skills that I had grown and had diversified, and I now work under the supervision of Dr. Gibran Kokar. So the Neurobiology of Dual Disorders Lab is focused on really understanding the neurobiology that underpins the association between substance use and schizophrenia and serious mental illness. And I, in particular, decided to join a very new lab. It was only 1 year old when I joined to gain insights about what it means to start a lab and what you can expect when doing that myself. So toward this end, toward the goals of the research lab, I'm going to highlight some of the data I've already produced now and kind of frame my research project now and hopefully into the future. So my current research focuses on detecting changes in neural circuit oscillatory activity that results from exposure to cannabinoids, and ultimately how these changes relate to changes that are observed in patients with schizophrenia. I intend to determine whether some of the dysfunctions we see in these oscillatory patterns are a driving force. Are they really a mechanism here through which cannabis can exert changes in otherwise healthy users and in patients with schizophrenia? Or is it a byproduct of other neurobiological aberrations that maybe we could use as a predictive biomarker of the prognosis for the disorder or for individual users of cannabis? So a little bit of background information on this point. Cannabis use disorder is diagnosed in approximately 51% of patients with schizophrenia. I've seen reports as great as 80%, and this is a lifetime rate of diagnosis for cannabis use disorder. And it is classified as a dual diagnosis as it presents unique clinical challenges for management and for treating. But unfortunately, only canonical or classic treatments exist. So classic treatments like antipsychotics or any kind of addiction therapy for cannabis use disorder. So there's really this urgent unmet need for managing this dual diagnosis effectively. And so we're focusing in on having a mechanistic understanding of the neurobiology with hopes it will reveal possible treatment targets as well as biomarkers. cannabis use, we know, worsens the overall prognosis for patients with schizophrenia through exacerbating symptoms, but also through things like medical noncompliance, so the inability for patients to adhere to treatment plans or to attend appointments, and through other socioeconomic factors as well. So it really begs the question, why then do patients use substances, including cannabis, at such high rates if really we have these negative consequences? And what else here is at play? So the self-medicating hypothesis is one of the leading hypotheses for the association between cannabis use and schizophrenia, which postulates that patients are using cannabis as a form of self-medicating, as oftentimes you'll see an acute improvement or amelioration of symptoms shortly after patients use cannabis. And so this is kind of exemplified on the slide here, showing that we have seen increases in attention, executive function, verbal memory when comparing patients with schizophrenia who are using cannabis versus patients who are not using cannabis, either chronically or acutely. But at the same time, we also see a large amount of evidence that supports the idea that cannabis use can negatively impact the symptoms of schizophrenia. Things like increase or decreasing working memory, decreasing sensory filtering, or sensory processing for patients. So ultimately, there's still much more to discover. What we do know at this time is that for patients with schizophrenia, cannabis use worsens the prognosis and creates treatment complications that we must address, particularly at this time when we do see recreational cannabis becoming legalized or decriminalized in Canada, especially. It has happened recently, and these are at-risk patient populations, and we must create solutions for them. Cannabis also produces effects that are similar to psychotic states. These are the psychotomimetic effects of THC and include states of paranoia, delusions, hallucinations, restlessness, and anxiety. Looking at some of the clinical data around this, there is a seminal paper, a well-cited study by Dr. Deepak Cyril D'Souza out of Yale University that was published in 2004. They found that after intravenous injection of THC in humans, they reported a transient increase in the scores of positive and negative symptoms. So as you see on the slide here, we have on the y-axis time in minutes before and after the infusion of THC. On the y-axis, we have the symptom scale, so positive and negative symptom scale reported scores, so it's PANSS. And this is a tool that's often used to diagnose schizophrenia. And so the graphs are depicting a transient increase in these scores after THC is infused that lasts or persists for around 2 to 3 hours after the exposure to THC. So importantly, this work was the first kind of clinical study to directly associate the psychotomimetic effects of THC with the symptoms of schizophrenia through this diagnostic tool. And it's been reproduced using other psychiatric symptom assessments. And I will touch on the link between these results and the brain's electrical activity shortly. But before I do that, I'm going to just provide a brief primer on brain electrical activity so we're all on the same page. So I think most who are interested in neuroscience and pursuing a career maybe in neuroscience and neuroscience research understands the electrical properties of the brain and of the neurons transmitting signals. So we're able to use recording techniques to pick up on those signals. A good example of this is EEG, of course, electroencephalography, that is used to pick up signal leaking through the cranium in humans. We also use in rats implanted electrodes, so wires that are able to pick up on the brain signal at deeper brain structures. And so the property of electricity and being on the part of electromagnetic, I guess, is that it functions like a wave. And so on this slide, you can see in the top left, we have an example of neurons firing or spiking over time, which is the x-axis. And you see there's this kind of phasic spiking, which then maps to this oscillating activity as represented on the graph or the figure below that, titled local field potential. And these oscillations create frequencies, of course, being the number of peaks over time. And so we have a number of different frequency bands within the brain that's depicted by the figure on the right, delta, theta, alpha, beta, and gamma ranges of frequencies. And these also are seen as cognitively relevant as they have certain functions they're associated with or certain tasks that they are associated with. Importantly, we can also use measures of power, so kind of the strength of the signal, and coherence, so the synchronization of different signals between brain regions or the same frequencies within or between brain regions to really assess how strong and functioning these signals are and whether or not there's any dysfunctional behavior in certain disease states or disordered states. So coming back to the evidence of the effect of THC or cannabinoids on the positive and negative symptoms of schizophrenia and psychosis, a paper published by Cortez Spirones et al. In 2015, who is actually in Dr. D'Souza's lab or in an affiliated lab, maybe Patrick Skosnick's lab at Yale. In this paper, they examined the effect of intravenous THC again on the scores on the PANSS tool or PANSS scale, but they also measured EEG and to detect whether or not there was any change in oscillatory power and coherence. And long story short, what they found here is that after THC administration, they saw a decrease in power and coherence, particularly in the gamma frequency range. So this range being from about 30 hertz to 100 hertz. And so importantly, on the right on this slide here, you'll see that when they performed a correlative analysis between the changes in brain signal and the PANSS scores, they saw that there was a negative correlation. So as the THC induced an increase in the positive and negative symptoms on the PANSS scoring, they also saw a decrease in power and coherence in this gamma frequency range. So this brings me to my current project, which is to explore the involvement of oscillatory disruptions through the suppression following THC in a rat model of schizophrenia. So there are 3 aims here. Apologies for the text on the slide, but I'll go quickly through them, so no need to read. Aim 1, we're looking at the impact of cannabis vapor exposure in healthy rats on their behavior and on neurophysiological correlates of behavior or their oscillating activity. Aim 2, we are exploring the impact of cannabis vapor exposure on schizophrenia-like behavior, so using a rat model of schizophrenia as well as the oscillations we are recording from, and we're recording from in different cortical limbic brain regions. And then finally, aim 3 will be to see what effect modulating that oscillatory activity may have on the schizophrenia-like behaviors, as well as the oscillations themselves over time, and whether or not modulating the oscillations or these frequencies could impact the effect of cannabinoid exposure on these measures as well. So to start, I'm just going to describe the protocols we are currently using in the lab towards those aims. First off, we are using a vaporized cannabis administration protocol. This is a protocol we have developed in-house. So these photos are of the setup that we have built. We have provided the protocol and the schematics open. They're open source or open access. You can see them in eNeuro. And I'm happy to provide anyone with that information if they'd like to reach out and build something like this in their own lab. But importantly here as well, there are 2 reasons why we're choosing cannabis vapor in this case. First off, it's practical. The onset following THC or cannabis vapor is quite rapid after inhalation, and the administration is quite easy to perform. In this case, we have off-the-shelf cannabis vaporizers purchased in Canada. We have cannabis purchased from the dispensary, and then we can use the cannabis in that vaporizer, hook it up to our vaporization chambers here, and ultimately administer the vapor to the rats in the chambers. And so it's very, very translational. And this brings me to the second point, which is that cannabis vaporization at large is becoming a preferred method or method of choice for many users. You're seeing an uptick, particularly in youth who are using cannabis. And so we're really trying to get at this highly translationally relevant protocol or platform with this administration route. Second, we are using the NVHL or neonatal ventral hippocampal lesion rat model of schizophrenia. This animal model is very well characterized in the literature. It has been shown to have face validity, predictive validity, and construct validity. So ticking the boxes in terms of a valid model for human schizophrenia, and it's relatively easy to create, and ultimately we have extensive familiarity in the lab with this model. So just briefly, The creation of this model involves lesioning with ibotenic acid the ventral hippocampus in rat pups when they are 7 days old. And then when they grow up into adulthood, they have or appear to have behaviors that are very much like the symptoms of schizophrenia. So on this slide on the left, you see this timeline of development on rat postnatal day 7, which is the top line. You can see we have the bilateral ventral hippocampal lesion with ibotenic acid. And then over time through up into young adulthood, the appearance of the cognitive negative and positive like symptoms will appear. And so we will perform all of our studies when the rats are in adulthood. The next protocol I will highlight is our recordings, our electrophysiology protocol. So we're choosing to do local field potential recordings. So we'll be recording the summed activity of neurons or networks of neurons in particular brain regions while the animals are awake and behaving. This is kind of comparable to EEG in humans, but we're obviously able to go into deeper brain structures as we can do invasive surgeries. So importantly, the animals are implanted when they're kind of adults. So we don't have to worry about any kind of developmental issues either with the brain still growing. We target it in, I'll explain the brain regions in a second actually, but we target a number of different brain regions with different electrodes, recording wires that will then transmit the signal to a digitizer and an amplifier to be processed and analyzed and transformed. And so the two behavioral tasks that I'm going to describe today as well, first one is acoustic startle response test to test sensory motor gating. And the second one I'll get to in a moment is the object recognition task. So acoustic startle response task is a well-characterized tool for assessing sensory filtering. So the filtering of incoming sensory information, and it kind of has hallmark deficits in rat models of schizophrenia. And so it functions on the principle that when you present a startle stimulus, so a stimulus that will elicit a startle reflex to the animal, you'll be able to record their startle response. And if you present a pre-pulse, so this is a sub-threshold stimulus, so something that not necessarily will elicit a startle response, in advance of that startle response, you will see that their startle amplitude or their reflexiveness or the reaction to the startle response is dampened. And so we have this pre-pulse inhibiting the startle response. And so I like to liken this to the brain hearing lots of noise coming from one area and ultimately saying, okay, I have noise coming from this area. I'm going to dampen my responsiveness to it so that I don't continually startle to this noise. And again, this is a deficit that you see quite clearly in rodent models of schizophrenia, as well as patients with schizophrenia. And so it involves just placing the animal in this box, presenting the different trials with either the startle only stimulus or the pre-pulse and startle stimuli, and recording their locomotor response to those stimuli. Okay, so the spontaneous object recognition task is a well-used, well-characterized task to assess memory. In the sample phase of the protocol, we will place the animal into a box where there are two identical objects on one side of the box. The animal will familiarize themselves with the objects, and then they'll be removed from the box for a period of time. In our case, we're using 5 minutes, so very short term, or 24 hours more long term, and we place the animals back into their choice box, where there will also be a new novel object. And this is built on the understanding that rats prefer novelty in the environment, or will at least explore novelty within their environment. And so in some way, it's a proxy measure of their familiarity with the, in this case, the yellow mug, and to spend more time exploring that, then we know that they recognize that as a novel object, and they must then recognize the soccer ball as a familiar object. And we calculate their time spent exploring either object as a function of their total time spent exploring. Okay, so now that we're through with the methods overviews, I'll dive into some of my data. So for AIM-1, looking at cannabis vapor exposure in healthy rats on behavior and electrophysiological measures, we used 3 protocols so far. The vapor admin protocol, of course, as well as our LFP protocol to record neurons, and then the acoustic or startle response task or paradigm. So I'm going to present some published data as well as some unpublished data. And so stay tuned for the final version of the unpublished data, and I'm happy to share that info around as well. And for This study here, the LFP study on the screen, it is published, and I'll show that publication momentarily. So we administered in otherwise healthy rats THC or vehicle, and in a crossover design. So in week 1, they received administration of THC vapor. Well, half of them did, and the other half received vehicle vapor. And then in week 2, it was reversed. So any rats that received THC in week 1 ultimately got vehicle in week 2. And likewise, any rats that got vehicle in week 1 got THC in week 2. And so it allowed for us to do some within subject comparisons. So on the screen, you'll see a number of figures. On the top row, we have the power of the brain oscillations based on brain regions. So we examined the dorsal striatum on the left column, orbitofrontal cortex on the middle column, and prefrontal cortex on the right column. On the x-axis for these graphs, we have their frequencies. So you can see it's just their power laid out against their frequency. And importantly, the big takeaway here is that compared to the vehicle values, so when they were just administered vehicle vapor, either week 1 or week 2, you can see that for the THC treatment, there's kind of suppression of their power, particularly from around 30 hertz through to 100 hertz. So this gamma frequency range. And this is just quantified in the bottom row where we have power spectral density, or the power for the gamma signal. And we're comparing vehicle to THC treatments, and we're parsing out low versus high gamma. But ultimately, we're seeing suppression across all gamma. And one figure I haven't included in the slide here, but I will mention now, and I do encourage anyone to check out the paper if they're interested in diving deeper into some of the other data that we produced, is that because we did this within subject comparison, we were able to look at the group that was treated with THC in week 1 and then vehicle in week 2. And what we saw in their brain activity is that this suppression, this gamma suppression, actually lasted, so that when we recorded them during their vehicle or after their vehicle exposure, they still had gamma suppression from the THC exposure a week prior. So this really indicates to us that although some of the behavioral effects and maybe some of the acute psychotic, psychoactive effects of THC may have dissipated, we still have this lasting brain suppression and across all brain regions, which is quite striking to us, and certainly calls to attention a need to really consider or maybe reconsider the washout periods that we have for crossover design studies. So it was published in the Canadian Journal of Addiction in 2019. And just as an aside, we also were fortunate enough to publish a review paper to the Journal of Pharmacology and Therapeutics, which you can access using that PMID number on the screen, which provides a detailed update on the preclinical research exploring how neurotransmitter receptor expression and behavior changes from adolescence through to adulthood and after exposure to substances like alcohol, nicotine, cannabis, and opioids. Okay, so now I'm going to present some of the unpublished data, and so the rest of the data in this talk will be unpublished. So yeah, stay tuned. So for PPI in the otherwise healthy animals, so not the rat model of schizophrenia, we ran a separate study where we used cannabis flower vapor instead of THC isolated. We purchased this at a dispensary, and we had one strain with CBD and THC in it, and another with only THC, or no CBD, I should say, in it. So we were trying to do a couple things here. Of course, look at what effect cannabis flower vapor would have on brain oscillatory activity, but also look at what effect the presence of CBD in that cannabis flower, so naturally occurring CBD, would have on changes in oscillatory activity. So we exposed the rats to 4 different pre-pulse levels, as you'll see in the x-axis here. So we have our conditions being no drug, which was their baseline values. Then THC was exposure to the cannabis strain without CBD. And then THC and CBD is exposure to the cannabis strain with both equal parts THC and CBD. Now, with that, we also see 76, 79, 82, and 85. Those are those pre-pulse levels, so they're decibel levels. And I realize I don't have the units. I'm sorry for that. So there we presented a 76 decibel pre-pulse stimulus before our startle stimulus. And typically what you'll see is the pattern that we have here now, where as you increase the decibel level or intensity for the pre-pulse, you see an increase in their percent PPI approaching 100%, where you'll have a truly like startle eliciting stimulus. And so what we're seeing here is that a couple things. We did have a significant effect of drug, but unfortunately, looking at the behavioral data, we're not confident that our protocol is working 100%. I mean, looking at the no drug condition for percent PPI, and again, these aren't manipulated animals. These are otherwise healthy animals. That's a really low PPI value. You'd expect something closer to like 80% or 0.8 in this graph. So it might be that there's a need for us to revisit how we're habituating our animals and the environmental parameters we're using for our protocol. So in that regard, if there is anybody on the call today who is familiar with the acoustic startle response paradigm, I'd be more than happy to connect with you as well to learn from you as we work to refine our protocol. Okay, so I'm going to now talk about some data that I've produced toward my second aim, which is to use a similar methodology, but looking at it in our rat model of schizophrenia. So in this case, we produced we used the MVHL rat. We are using cannabis vapor administration, our LFP recordings again, as well as the acoustic startle response paradigm and our spontaneous object recognition, or novel object recognition as it's called. Again, all of this data is unpublished, and this is brand new data here. So it's kind of first time it's being revealed or presented. So enjoy, and please bear with me. I know it's hard to see on the screen, and I'm sorry, this is the only version I could receive from the collaborators we had that were using this data. So I'll just provide a general takeaway, which is that we saw very few baseline differences in the oscillatory activity between NVHLs and shams. Sham surgery controls. So the MVHLs received their ibotenic acid lesion. The sham group went through the procedure, the surgical procedure, but we only administered artificial cerebrospinal fluid, and we did not administer ibotenic acid. So it's a sham surgery. There isn't any lesioning. And so we saw between MVHLs and shams at baseline that there was a suppression or decrease in gamma in the cingulate cortex for NVHL rats. So they already had this kind of baseline deficit, which mirrors what we do see in clinical studies for patients with schizophrenia, that there are baseline deficits, although the picture is a little murkier with the clinical data because some areas have increases or enhancements and some have reductions. After cannabis exposure, again, general takeaway here, we saw lots of suppression of oscillatory power in many different frequencies in many different brain regions, mostly the similar patterns between NVHLs and shams, with the major difference really being that for the cingulate cortex in that gamma frequency range, we saw that THC and CBD strains of cannabis have further reduced that already present deficit more so than we saw in the sham. So maybe this is indicating to us that there's some sort of two-hit process or two-hit factor where, because there's already deficits in their baseline signal, the exposure to cannabinoids or cannabis can ultimately worsen it further. We're not really sure yet. As I said, this is brand new data, and so we're kind of working through what all of this means presently. This is, again, just to show that we saw it across multiple brain regions. And when, additionally, when comparing the THC only or the cannabis strain without CBD to the cannabis strain with CBD, we saw something really interesting, which is that the cannabis strain with THC and CBD seemed to have an ameliorative effect. So the suppression was to a lesser extent than was observed with the cannabis strain that did not have CBD. So we're seeming to reproduce results that have been shown in recent papers, one out of the La Violette lab here in Canada, as well as a few other papers showing that CBD has the ability to kind of counter the effects of THC. And again, more data is coming out of this, so stay tuned as we work to get it published. Okay, so we did run PPI again in this cohort of animals, and we did see some better values than we saw in the other group I showed you earlier. This time we used the MVHLs and shams rats, and we used cannabis flower vapor, not THC vapor. So we overall saw an effect of surgery. It was non-significant effect when we controlled for outliers and some variability in the data. But just looking at the figure on the screen here, you could see on x-axis we have our sham groups on the first 4 ticks, and on the last 4 is our MVHL group. Again, we have our pre-pulse levels at 76, 79, 82, 85. On the y-axis is our percent PPI. And just looking at the sham and MVHLs, you can see the MVHLs have this deficit in percent PPI compared to shams. Again, that was not significant, but it was very close. We did not see a main effect of drug, and this was also very close. likely due to maybe statistical power. Our sample sizes were very low once we did remove some outliers. So please, again, this is a call to anybody, a call to action on the call. If you're familiar with PPI, if you're familiar with the acoustic startle response, please reach out. I would love to talk with you more. Okay, so finally, we are starting to run our spontaneous object recognition task in a separate cohort of MVHLs and sham rats to see if we can detect any, well, baseline behavioral changes and ultimately any changes resulting from cannabinoid or cannabis exposure. So I have only the baseline data here for you today. We have yet to run the cannabis administration portion, but what we have here is a very clear deficit that is delay dependent. So the MVHLs and sham rats performed similarly in object recognition tasks when there was the 5-minute delay. So they both kind of explored and could remember their familiar object and explored their novel object at the same level or same amount. Whereas when we did the 24-hour delay, so the rats had 24 hours before they needed to recall what the objects were that they saw 24 hours prior, we see that there is this deficit whereby the MVHLs really could not recall the familiar object, again, as a proxy measures through their time spent exploring the novel object, which comes with a whole host of assumptions and interpretations that I'm happy to discuss as well. But we're really happy to see this kind of baseline deficit, this delay-dependent deficit, and I think it'd be really interesting to see ultimately what effect cannabinoid exposure will have on this. Okay, so overall, just to synthesize where we're at currently and ultimately provide some insights into where I'm hopefully going with this, we were able to demonstrate through a couple cohorts that cannabinoid exposure acutely disrupts oscillatory activity, and importantly, that these disruptions seem to last at least 1 week. So it would be interesting to explore, of course, how long they last beyond 1 week, looking at 2-week washout periods and looking at electrical, physiological activity after that. We've also concluded that our behavioral protocols and setups can be quite challenging, particularly when we're using the cannabis flower vapor, I think, because there's a lot of contributions of the other constituents in terms of confounding some of the results that we're getting. But we do have clear deficits from surgery as baseline deficits. So we just need to refine our protocols a little bit, refine our cannabis administration a little bit. Importantly, we are seeing this bidirectional response between THC and CBD in that we saw a suppression through the cannabis without CBD and an amelioration when CBD was present. So getting at this idea of this duality that exists within the mechanism of actions for psychoactive substances, but also I think at a higher level, it's interesting to reflect on the duality of the acceptance of these substances as well. So before I get into that, I'd like to just say maybe the self-medicating hypothesis I had described before really describes the use of cannabis by patients who are seeking the benefit of CBD and ultimately are ingesting THC as a byproduct of it being in the plant along with CBD, and then entertaining the negative consequences from that as well as the positive. So it could be that the self-medication hypothesis has value in explaining this relationship, but ultimately is not it doesn't indicate causality at all. So I like to consider if we can encourage patients to use cannabis with a minimal amount of consequence. Is there a way we could promote a certain amount of CBD in the cannabis that they are going to use, like they did with patients with epilepsy, where they're promoting the, I think, Charlotte's Web strain of cannabis. Also, and my next step for this is going to be see if we can modulate oscillatory activity in a way that maybe reduces cannabis use, cannabis dependency, or maybe some of the symptoms of dual diagnosis. And again, coming back to this point of duality, can we really leverage this fact that we have, on an upper end, maybe some negative consequences of psychoactive substances, on the lower end, maybe some really incredible benefits from them, to finding a sweet spot where we can have users continue while we're minimizing harm. And I think a good example of this is kind of the microdosing push now with psilocybin and with lysergic acid, where people are taking small amounts and reporting benefits, but not necessarily getting some of the kind of more psychedelic psychoactive effects. Like hallucinations and whatnot. So importantly, I think we really need to consider how we intend to use, or what is our intent with using these substances, so that we can benefit from some inherent pharmacology that exists with these plants and that we've kind of had under embargo before. And I'll just take a moment to stand on my soapbox and say, as scientists, particularly scientists in psychoactive substance research, we need to be careful not to bias public opinion. There's lots of opinions about psychoactive substances, and so we just need to be really vigilant to kind of challenge our own assumptions about what they can do, what they mean, and what impact they'll have on society. So I'm about to wrap up, a couple slides left here. First, the future directions. As mentioned, my next aim will be to explore modulating gamma signal and see what effect that has on schizophrenia-like behaviors in the MVHL rat, as well as any changes in oscillatory activity over time, and then to see if that has any impact on how cannabis impacts the symptoms of schizophrenia. I'd also like to maybe possibly explore the role that glial cells play in mediating the impact of cannabinoids on behavior and these neurophysiological correlates. And importantly, the significance for this work is kind of twofold. Overall, we're looking for new measures to understand the relationship between cannabis use and schizophrenia at that neurobiological level. But we're also hoping to identify biomarkers possibly through these changes in oscillatory activity, or maybe even a means or targets for stimulation-based interventions. Previously cannabis was kind of thought to be causal, or at the very least like a strong risk factor, although it still is presently for developing schizophrenia. Now we're seeing some evidence that schizophrenia is also causal to the use of cannabis over time. So if you're predisposed towards schizophrenia, it's likely that you're going to use cannabis in your lifetime. So again, there seems to be this bidirectional causal relationship, and it's likely then that this may be indicating some sort of causal mechanism that underpins both of these. And we're thinking it's going to be circuit dysfunction, either driven by the changes in oscillation or oscillatory activity, or that is then reflected in these changes. Finally, as a kind of future next step, I hope to finish up my PhD and ultimately pursue other interesting lines of inquiry, namely looking at how psilocybin could be used to assist with withdrawal for patients with schizophrenia. One problem with their substance use is that they experience a drastic and very impactful withdrawal, and often then take to using other substances as a form of managing that, as do other patients with substance use disorders. And so I think it'd be an interesting use case for psilocybin. We're already seeing some positive results from clinical studies. Likewise, I'd really like to maybe look at a future kind of moonshot project of how we could maybe look at therapeutic uses of mescaline. Although this is not my forte, I'm not well versed in this area at all. I just think it'd be a really cool and interesting avenue for me to pursue. So with that, I'd like to shout out everybody in our lab. This is the current and past team. I'm not going to run through the list of names, but we are a wonderful family, and I encourage anyone interested in following up or keeping in touch with us to just check out the cocarlab.com website. I'd also like to acknowledge my department at the Ontario Veterinary College, so the Department of Biomedical Sciences here at the University of Guelph, for the scholarships and support I've received to complete this research. And finally, let's connect. I have a different experience, I think, in academia and beyond academia, and I'm happy to chat always about my experience in life, my research, and my passions. So feel free to connect on any one of these platforms. And thanks so much again for the opportunity.
Okay, thank you very much, Bryan. We sure enjoyed all your exposition, and we're glad for you share with us all your experience and your research too. So we have some questions about your public. Thank you very much all you are connected on this webinar with you. And I start with the first question. Bryan, the first one. What are the ethical considerations for the use of the opiates in the research you have? I would copy you the question in your chat too.
Okay, thank you. What are the ethical considerations for the use of opiates in research? So it's a broad question, and I'm glad that one of the first questions is around the ethical considerations, because this is important and arguably should always be the first questions in many different sectors. I'm going to speak or try and answer this from the perspective of a preclinical or animal researcher. Importantly, considering the subject's well-being, so in my case, animals' well-being, is first and foremost. So there are many regulations and many committees and many safety checks that are inherent in the process of getting approvals to use these subjects in research, these animals in research, that obviously take into consideration some of the ethical priorities around use, things like making sure that the pain or the risk for negative health consequences that you impart on subjects is minimized as much as possible to not overuse animals. So the idea of really reusing animals and repurposing animals and only using the number of animals that you require for the statistical power of your study and not overusing these animals. And so I would say top priority here, I mean, of course, in animal research and beyond, is really to minimize the impact or harm that you place on others. In opioids and research, it's a required element of research. We need to be able to understand how opioids work and how ultimately they will impact human health and arguably health for animals as well. And so we just have to minimize the amount of risk that is imparted upon individuals through the use and study of opioids. I think we are all familiar with, or maybe most of us are familiar with, the problems that have happened in the United States and in some other areas. We certainly felt it as well in Canada, where we had this opioid crisis, where there was essentially way too much being flooded into the market, overprescription or overprescribing of these compounds without really considering the long-term societal consequences and public health consequences of this. And so, I mean, now we're seeing a change for it, but unfortunately that's led to generations being addicted and priming many individuals for using other substances as a means to alleviate the withdrawal effects they'll see from opioids. So I think that was a perfect example of where some of the ethical considerations maybe were not prioritized. And unfortunately, that has had some negative health consequences. But I think, again, from my perspective, just considering the minimization of harm and the possibility of reusing subjects where necessary will help to ensure that there's a minimal disruption and we can still get some good results. And I'm happy to chat more at length about that. Please reach out.
Thank you very much. Thank you very much. Another question. As your neuroscientist, would you recommend cannabis treatment for personality disorders?
So it's an interesting question, and I'm happy to provide insights on it. I, as a neuroscientist, cannot recommend any form of treatment. I am not a medical doctor, and so I cannot speak to it as a treatment per se. Especially with personality disorders where there is a risk for a negative consequence with cannabis use. It may be negatively impactful for individuals to use cannabis as a treatment. But at the same time, the picture is very unclear right now. And so there are instances, much like the instances I highlighted with schizophrenia, where there are reported improvements in symptoms, and also, of course, the negative consequences. And so what I think is going to be required is a concerted effort for us to understand the constituents of the cannabis plant and how different compositions of those constituents or different strains of cannabis impart the positive and or negative benefits for otherwise healthy users, as well as for individuals with serious mental illness or other health or considerations. So I would not recommend cannabis treatment solely from the fact that I'm a neuroscientist and I am not well equipped to do so. I think it's very much worth investigating both the positive and negatives of using this as a treatment for personality disorders.
Thank you very much. Also, your question. The next question. Can we infer that the same animal used for the experiments with THC or other substance were having all hallucination during the administration of the substance?
Yeah, this is really, really cool question. Thanks so much. So I think the classic answer is no. We cannot ask the animal if it is experiencing a hallucination, any kind of psychedelic side effect. And so it's really hard for us to be able to say whether or not they are hallucinating. So most would say, no, you cannot. There are recent studies that are making the case that we can use certain behaviors, like unnatural behaviors that the animals will exhibit after administration of psychoactive substances, as a proxy measure of hallucinations. But again, we will never close that gap because we can't ask the animal directly. So I think we are working as a field toward a measure of hallucinations in animals, but we will never be able to close that gap. importantly, what we can do is compare brain activity during those periods that we think may be hallucinations and compare that to human data and see if there are similarities in the way the brain is responding, maybe using some MRI data to determine if we could realistically say it's a hallucination or not. But again, we won't close that gap.
Thank you very much. Another question. What are the prominent strange variables that you have to the control when experimented with animals? That's a usual problem.
Excellent question. And again, you'll see with my presentation of my PPI data really how variable the results can be and how many considerations are required before we can get clear, crisp data. So prominent strange variables, I would say, in animal research are mostly born out of environmental parameters. So things like the time of day, the light condition in the room, the temperature of the room, the odors in the room, the previous handling of the animals. These are all things we can control, of course, but there are also things that are very hard to predict how they will impact your research and how they will impact your results. You know, we, for the longest time, had a set of, or a group of parrots on our floor in the research lab for a group that was researching parrots, and they are very noisy. And so that ambient noise itself can ultimately impact the results, especially with PPI, where it's an acoustic based test or task. So beyond that, I would say prominent strange variables would be inherent differences in the animals themselves. We've noticed that animals often have their own kind of characteristic, their own personality, if you will, not to personify the animals, but they kind of have their own performances. And so we need to also control for those unique characteristics when we're, say, assigning them to treatment groups or when we're interpreting their behavioral data. Okay. One other interesting thing as well, I just expand on quickly, for anybody who's interested in doing the cannabis vapor administration protocols, we're really seeing an interesting result in how the animals respond to the cannabis vapor behaviorally, depending on their social dominance. So if they're if we have them pair housed and they're the dominant rodent, they are often still very alert, even though clearly there's a behavioral effect happening because the submissive rodent is kind of clearly inebriated as a result of the cannabis administration, but the other one is very alert and almost protective of the submissive. So I think getting into looking at this naturalistic behavior as a result of cannabinoid exposure will be a really interesting area to pursue.
And you use some control of the family, son, mother, or brothers with your mouse, or that thing. How do you do that thing?
Yeah, great question. So because the NVHL rats specifically, we receive them as a litter. So we receive them with their mums because they're only 5 days old at the time that we receive them. So we know who birthed or what rat birthed them. We know which were litter mates. And so we can keep all of them together to really decrease any confounds from social stress or maternal separation stress. And so for that, it's quite easy to work backward. For the animals that we order in as adolescents or as adults, it usually just comes down to the ordering process. And it's less of a concern once they are kind of fully developed for us.
Okay, awesome. Thank you very much. Another question. Does the drug have cumulative effects with the repeat administration?
Excellent question. So there is a steady state where you will have saturated the system. And so there will be kind of, so far as I understand it, cumulative effects up to a point at which you will be saturated. And this is highly variable depending on the administration route you choose. So if it's inhaled or injected, it's often going directly to your bloodstream and to the brain, and it bypasses any first pass effect that you would get with metabolizing, say, an edible if you were to eat that and digest it. And actually eating edibles and digesting that creates byproducts that are often even more psychoactive than THC itself. So there are cumulative effects, but it's highly, highly variable, so far as I understand it, but it's highly, highly variable depending on the administration route, as well as, of course, the number of repeats and whatnot.
Okay, thank you very much. Another question. how can the results from animal models can be translated to humans? It's a nice question today.
Yes, it's a great question, and it's the ever-present problem with animal research. I think, of course, you know, on one hand, it's valuable to explore the rodents for understanding rodent behavior, but we in the preclinical space always use them as models for humans. So importantly, we just need to make sure that we are prioritizing the validity of the model, as I highlighted in the NVHL rat. So we should make sure that it is very similar, appears similar, the animal responds in a similar way, and you can see a lot of overlap in different measures to the human condition. We will never be able to say with any kind of level of like 100% certainty that this produces the human version, but we do see a large degree of overlap. A great example of this is any Alzheimer's model, where you see the plaques and the fibrils and whatnot. So we're able to produce human-esque disease pathology or disorders in these rats. So we can say with a degree of confidence, but again, we can't say 100%. For my results specifically, I would love, I mean, we can't do the deep brain structure, so we're never going to be able to, well, I shouldn't say never, but right now we cannot implant humans with these far-reaching electrodes and record from those deep limbic brain structures. So we're kind of lost there. But I think we can look at changes in oscillations using EEG and using a number of different cognitive tasks during cannabis administration before and after as well. And this has been done in the past, but because cannabis as a substance has been illegal in many places for a long time, the research has not really been allowed to pursue or to grow. And so there's a huge, huge gap, even in the clinical data, and especially in the preclinical data, that we can take a lot of the results from preclinical, build a clinical study around it, and ultimately then test. Another important thing I'd like to highlight here in this question is reverse translating results from clinical data to preclinical data is really helpful in this area right now because, for example, we don't have a strong model of self-administration or of cannabis dependence yet. So we've seen cannabis dependence in humans. We don't have that yet in rats or in rodents or anything. So reverse translating those findings into preclinical will help us create a solid foundation in the preclinical space.
Okay, thank you very much. That's a very, very important know about that and the use, how to use to research about this kind of intervention or this knowledge. Another question. Can these substances that can affect the memory be able to specifically change negative memories?
Interesting, very interesting question. So the short answer for that, I would say, and I can say this relatively confidently, is yes, there is good evidence that substances that ultimately affect memory and create different brain states can be used to change negative memories. A wonderful example of this, and arguably one of the leading kind of psychoactive investigations for therapeutic purposes, is with MDMA and individuals who are experiencing post-traumatic stress disorder. So oftentimes these substances can create a state in the individual where they're very reflective, but they do so almost objectively or of external of any kind of personal bias, external of any kind of emotional attachment to those memories, to those experiences, which allows for, through associated behavioral therapy, transforming those negative memories into something different or something that's not as impactful negatively. So you see this a lot with changing. We have really good evidence actually of changing the salience that an animal will attribute to an object in the object recognition task. And so if anybody's interested in that, the Winters lab, Boyer-Winters at the University of Guelph, has produced a lot in terms of understanding how rats change the context that they associate with a memory, and exposing those animals to substances like MDMA that create that state where you can reflect without that contextual association, then of course opens the door for helping to move forward from that. So I would say yes. Can cannabis do it? I wouldn't say no. I haven't personally come across any evidence where it has created a state of that nature. It's hard to get around some of the negative consequences as well, like the paranoia that may come with it. And so I'm not sure cannabis is the best tool for it, but would be a great avenue to investigate.
Okay, thank you very much. Near on the end, where do you see these files of study evolving over the next few years?
Great. Excellent question. So this is my opinion, of course. I'm still learning a lot about the field as well, but I see there being a couple main challenges for preclinical research, preclinical cannabis research. Number one, really figuring out the differences between the administration routes. So I highlighted in my study, in my talk today, that we used isolated THC. So this was THC suspended in a vehicle that we then administered using vapor. We also used cannabis flower vapor. So this was just the plant that was vaporized. And so we saw drastic effects across those two. I mean, we had in one case isolated the THC constituent or molecule. And on the other hand, we had all of the phytocannabinoids and other terpenes and whatnot associated with it. So there's definitely differences in the substance that you use or the form of substance that you use, but also again, highlighting the different administration routes, like the first pass effect with metabolizing it or not. So we need to basically figure out what each of those administration routes and doses and compounds or forms will impart on the user. And then beyond that as well, another area we need to focus on now is creating a reproducible and reliable model for cannabis dependence, because we have the data showing that there's cannabis addiction and cannabis dependence. We just have not been able to produce it in a rat model yet. And so that will be, we're on the precipice of getting that now. I think in our lab is something we're working on closely. Where do I see it evolving in a few years? It's at a high level, it's going to be really interesting because we, in Canada especially, but in other areas now as well, we're having these industries form around cannabis. And so we're ultimately going to be getting a lot of societal data that will inform our research. I think that will be really interesting to kind of blur that line, and reverse translation will become imperative at that point. Also, using deep learning, machine learning techniques, AI techniques, kind of high computing processing techniques to be able to really understand the constituents in cannabis, like what's each one of these constituents impact, because it's a large amount of data that we'll get with the many different strains of cannabis that exist out there, and we're going to require some high computing power to process everything. So I think watching how technology will impact this field as well will be important. Yeah, I'll leave it there, and I'm happy to chat again more at length about that. It's a really cool question.
Yeah, sure. And the last one, what do you think is the evolution that we should be looking for regarding to humans?
Interesting. Sorry, evolution. Is that what you had said?
Yeah.
What do you think is the evolution we should be looking for to humans? Interesting. Okay. Could I clarify with whoever asked the question what they mean by evolution? I'm thinking of it from like a evolutionary biology.
Yeah, I think it's for the next research, for the next something to look for. That's the kind of the research of the neuroscience. And
great. So I would say for cannabis research, really looking again at the different constituent compositions and how those can be used therapeutically, and how maybe some of them should not be used therapeutically will be very helpful. Again, a good example of this is the Charlotte's Web strain of cannabis that they use for epilepsy solely. And I think it has like FDA approval and everything. So figuring out what other use cases we have with cannabis will be really, really important for humans. I also think that there's a lot of merit in exploring the changes in electrical activity, of course, because it's my interest, but also because we're seeing some really interesting data from that. And there really hasn't been much data in that field at all yet. Importantly, looking at patients with serious mental illness, we're going to need to start considering poly-substance use. So the idea that they're not using just cannabis, they're using cannabis and nicotine and alcohol and opiates and other substances all together. And really, what effect does that coagulation or kind of compounding of all of these substances have on health? And is there a way that we can reduce harm in that dynamic? Because there's a great deal of evidence also showing that the effects of each substance combined is much different than the effects in an individual sense. So I think that will be very important. Oh, and one final thing, withdrawal or cessation of use has not really been explored all that much in serious mental illness, or particularly in schizophrenia. We don't understand what effect withdrawal has on biology all that much, and how individuals ultimately end up taking up other substances that they use. And so controlling those factors will be really helpful for harm reduction and treatment and whatnot.
Okay, thank you very much, Bryan. It's awesome We have to hear you, and you can share with us all your experience or your research. We're very, very, very glad with you. So for the end, well, I think we have to come to the end, but first I would like to make some commercial break for all. This event was co-organized by the NeurotechX Student Club at the Bolivian Catholic University. If you are interested in discuss topics related to brain-computer interface, biohacking, psychology, and neurotech, this is the place for you. We will so issue a call so that all interested students can apply and be part of that club. This was also made possible with the experimental research unit from the psychology department of the same university. I'll come soon. We hope to come back soon with more thoughts from the experts on different areas of research. Thank you very much, Bryan. Really, thank you very much for being here. I hope that we can meet again with maybe more projects between hand. And thank you for people being here. ¿No? And bueno, lo digo en español. Muchísimas gracias a todos los que estuvieron presentes. Bueno, algunos problemitas técnicos como suelen pasar en este tipo de webinars. Pero bueno, esto es una organización de parte del Club de Neurotech de la Universidad Católica Boliviana y la Unidad de Investigación Experimental de la Universidad. Veo a muchos estudiantes con mucho agrado y mucho aprecio, ex estudiantes también que de seguro están mucho más arriba, y que bueno, creo que este es un camino real de la ciencia, de la psicología. Creo que Bryan es una inspiración para todos nosotros en todas las cosas que se pueden hacer en cuanto a la investigación y a la psicología como tal y a la neurociencia como tal, ¿no es cierto? Y creer que las cosas pues están más cerca de lo que uno cree. Muchas gracias, Bryan, por todo el compartir con nosotros. De seguro estaremos contigo en alguna investigación, espero, ¿no? Y en alguna otra oportunidad. Well, thank you very much. Really, really glad to be with you. And thank you all to be participating in this webinar. Thank you very much.
Thank you, everyone. Take care.
Thank you. Well, see ya.